In this video, we will address lipid catabolism and protein catabolism. Lipids and proteins are molecules that can be used by cells, in addition to carbohydrates, to meet their energy needs and generate precursor metabolites for anabolic reactions. We are going to address lipid catabolism first. The most common type of lipid utilized by cells to generate energy in the form of ATP and precursor metabolites are the triglycerides. Triglycerides are composed of a glycerol and 3 fatty acids, as seen in the top picture. Triglycerides may be separated into their glycerol and fatty acid components via a hydrolysis reaction. The reaction requires 3 water molecules and is catalyzed by an enzyme known as a lipase. The glycerol component released from the hydrolysis reaction can undergo further catabolism by entering glycolysis. In order to catabolize a glycerol molecule via glycolysis, the glycerol molecule is converted to a molecule of dihydroxyacetone phosphate by the addition of a phosphate derived from ATP. As previously seen in carbohydrate catabolism, the dihydroxyacetone phosphate is converted into glyceraldehyde 3-phosphate and is then oxidized to pyruvic acid in order to produce ATP. The fatty acids released from the hydrolysis reaction may also be catabolized further. The fatty acids are broken down through a process known as beta oxidation. During beta oxidation, enzymes break the carbon atoms of the fatty acid off in pairs that are joined to a coenzyme A, thereby producing acetyl-CoA molecules that may enter the citric acid cycle. Carbons containing hydrogen are removed 2 carbons at a time until all of the carbons of the fatty acid have been converted into acetyl-CoA. Beta oxidation will produce the reduced electron carriers NADH and FADH in addition to many molecules of acetyl-CoA for the citric acid cycle. Many organisms will utilize proteins to produce their energy and precursor metabolites only if they do not have access to carbohydrate or lipid sources. This is because proteins are important structurally and functionally within cells. Cells such as the pathogens we are studying this semester will catabolize proteins as an important source of energy and metabolites. When you have an infection with an organism such as a pathogenic bacteria or a pathogenic fungus, those organisms will catabolize your proteins to meet their cellular needs. In layman's terms, we can say they are eating your proteins to get their energy. Most proteins catabolized by such organisms are too large to pass through their cell membranes. Therefore, these organisms use exoenzymes to begin protein catabolism. Exoenzymes are enzymes secreted outside of the cell. The exoenzymes used to begin protein catabolism are known as proteases. Proteases act to break a protein down into its individual monomers, the amino acids, by breaking the peptide bonds that join them. The amino acids that are released from the action of proteases are then small enough to pass through the cell membrane into the cell. We will revisit exoenzymes in Unit 3 when we explore virulence factors in more detail. Once inside the cell, the amino acids may be further catabolized by removing the amino group in a process known as deamination. The molecules left after removing the amino groups resemble the carbohydrates found in the citric acid cycle, and therefore may act as substrates in the Krebs cycle to produce energy for the cell.